Hello students, today we're going to discuss two major ways in which the cells move their stuffs in and out, using small bubble-like structures called vesicles. These are part of the cell's endomembrane system, like a highway network inside the cell involving endoplasmic reticulum, Golgi apparatus, etc. First, exocytic secretory pathway. This is to send things outside through the plasma membrane. But what gets sent out? Proteins, like antibodies or enzymes. Lipids. Glycoproteins. Small molecules. Basically anything the cell wants to export. How does it work? 1. These things are made or processed mainly in the endoplasmic reticulum, then go to the Golgi apparatus. 2. In the trans-Golgi network, the exit side of Golgi, they get packed into special vesicles. 3. These vesicles travel to the plasma membrane. 4. The vesicle membrane fuses, joins, with the plasma membrane, the contents spill out, this is called exocytosis. 2 modes of secretion. Constitutive secretion, always on, like a tap that's always dripping. Happens continuously, non-stop. Example, plasma cells make antibodies, immunoglobulins, and keep releasing them steadily. No need for any signal. Regulated secretion, on demand, like a tap you open only when needed. The cell stores the stuff in big storage bags called secretory granules or vesicles. Nothing comes out until a signal arrives, for example, nerve signal, hormone. Example. Salivary glands store saliva enzymes release only when you smell food or autonomic nerves tell them to. Special case in polarized cells, cells with top and bottom sides, like skin or gut lining cells. Most epithelial cells release stuff only from the apical side, top side, facing outside or duct. Pancreas, into duct. Stomach lining onto surface. Liver cells. Bile goes out, apical side, into tiny bile canaliculus. Blood proteins, like albumin, go out basolateral side, bottom or side facing blood sinusoids. The cell knows where to send vesicles because special address tags sorting sequences are on the proteins in the vesicle membrane. Think of it like postal sorting. The Golgi puts the correct zip code, vesicle goes to right door, apical or basolateral. Second, endocytic pathway, just opposite the exocytic one. Here cell takes things inside from outside, through plasma membrane. This is important. To bring in nutrients, signals, hormones. Controls cell communication, signaling, by removing or recycling receptors. Helps in fighting infections. Used in medicines, some cancer drugs enter via endocytosis. Removes old or damaged membrane parts. How does it work? 1. Starts at the plasma membrane. 2. The membrane bends inward, forms a small pocket, pinches off, creates a vesicle inside the cell, endocytic vesicle. 3. This vesicle goes to early endosomes, sorting stations, then to late endosomes. And finally fuses with lysosomes, the cell's stomach, full of digestive enzymes or hydrolases. 6. Contents get broken down into small pieces, cell can reuse them. Modes of endocytosis. Clathrin-coated vesicles and cavili, common ways to start. Phagocytosis, cell eating. Big particles like bacteria, dead cells, pathogens. Done mainly by specialist cells like macrophages and neutrophils. They form phagosomes, fuse with many lysosomes, destroy the invader, for example, bacteria. Other lysosome jobs. Fuse with autophagosomes, eating damaged cell parts, autophagy. Fuse with phagosomes. Even help repair plasma membrane by fusing back. Simple analogy. Exocytosis means sending a parcel out. Endocytosis is receiving and opening a parcel, often breaks it down in lysosome, recycling center. That's all for today's lecture. I hope you like the video. Please subscribe this channel and press the bell icon.